right, so we're going to do the second part of DNA notes. Here we go. So we left off on this slide here. DNA has these 4 bases, A, T, G, and C. So remember, A only bonds with T, G only bonds with C. That's where we left off. We have those complementary pairs. Now, what about the rest of the DNA? Okay, so DNA is made with these 2 strands. So the 2 sides, 2 strands that are in the shape of a double helix. So we have the 2 strands, and then they are twisted up. Okay, so like you twisted it. And they have that double helix shape. And the only reason it's like twisted up and then compacted, so it takes up less space because it's actually very, very long. So the sides of the helix referred to as the backbone of the DNA. So here's the sides, right? Here's the sides of the DNA are made up of alternating sugar and phosphate molecules. You should have heard that in the video yesterday for the opener. It explained it pretty well. So if you haven't looked at it, I would look at it. The sugar in DNA is called deoxyribose. So like this, let's say this is a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. All down the sides, creating that backbone of the DNA. The type of sugar is specifically called deoxyribose. If you have not heard of a lot of sugar terms, like I know I've heard people who have diabetes, like they talk about their glucose levels, and it ends in ose, glucose. I know I've heard sucrose, fructose, lactose is a milk sugar. All of those are types of sugars. So they're bonds between carbons, hydrogens, and oxygens. It's just a type of sugar. And that sugar molecule, deoxyribose, is holding the backbone of the DNA together. It is also connected to a phosphate. Phosphate is PO4. So that means phosphate bonded to phosphorus bonded to 4 oxygens, and it has a -3 charge. That -3 charge. So phosphate, 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 having that -3 charge in each of those phosphates on the 2 sides of the backbone of DNA is become really important to remember. The fact that it has a negative charge is what makes the process that creates a DNA fingerprint work. If it didn't have a negative charge, the process wouldn't work. So keep that in mind. DNA has a slight negative charge because of those phosphate molecules holding the backbone together. The rungs of the helix, so double helix, so the rungs mean the things in the middle, are made up of paired nitrogen bases, which is what we talked about on the previous slide, the A, T, Gs, and Cs. So those paired nitrogen bases are in between, which help code the DNA for instructions for the cells. So every little segment of a section of your DNA is a gene that codes some kind of instruction for your body to make something that your body needs to function. So if we look at next slide, so here's our DNA double helix. So we have sugars and phosphates all down that backbone of the DNA. We have A's and T's in between. So those are the nitrogen bases, and they really look more like the A is this double molecule, T's and C's are the single molecule. The whole group of them is called a nucleotide, having the sugar and the phosphate and a base. One section of that is called a nucleotide, and those are all down the DNA strand. Types of DNA. DNA in chromosomes is called nuclear DNA. So it's in your nucleus, okay? Nuclear DNA is inherited from both the mother and the father and is virtually identical in all cells of the individual's body. So all 46 of your chromosomes, 23 from mom, 23 from dad, are in each of your nucleuses of your cells, except in your sex cells, which we mentioned in a previous slide. They only have half, because obviously half in the egg, half in the sperm creates one new full baby. So that's just kind of how our reproductive systems work. But all the rest of your cells have all 46 chromosomes inside the nucleus of all your cells. So mitochondrial DNA, though, so we have nuclear DNA, which is the regular one we mostly talk about. There is another kind. It's called mitochondrial DNA, and it's in the form of a circular loop. So it's not in a double helix, it's in a circular loop. And unlike nuclear DNA, so the normal one coming from both parents, it is only inherited from your mother. So we can only track it back to you and your mother's lineage. We couldn't track it to your dad's side. Sometimes mitochondrial DNA is brought up, and it will probably be brought up in at least one of the cases we'll talk about. It lasts longer in the body. It doesn't decay as fast, and it's in some of the things that last longer in our bodies. So sometimes if it's been a long time, so if you find a really deceased person, sometimes the only thing that's left is the mitochondrial DNA. Next one. So just kind of showing you. So you have your cell. All your body is made of cells. It has a mitochondria and that would have the mitochondrial DNA in it. And you have the nucleus, which has your nuclear DNA in it, which is all 46 pairs. Okay, genome. So the human genome is the total amount. I don't want to stop here. Okay, a few more slides. So in the human genome is the total amount of DNA in a cell. It is contained in both the nucleus and the mitochondria. The human genome consists of 3 billion base pairs, so there's like a whole bunch of combinations of genes. The purpose of the genome is to code the blueprint for the human body and how it works. It is responsible for telling the body to make proteins and other needed molecules, so it's a code. However, not all 3 billion bases are useful, okay? So there is a lot of things in our long code of DNA that doesn't seem to truly have that much of a purpose. So some of it seems really purposeful and not all of it does. So exons are encoded DNA with directions to build molecules. Okay, so it's like a specific section. These exons is a segment of your DNA that tells it to build a certain type of molecule. Exons make up only about 1.5% of the entire genome. Together, they code for about 24,000 different genes. Obviously, we don't have all 24,000, like that's just the possibilities that we could all get. The rest of the genome is made of introns. So introns are unencoded DNA that do not code for the production of a molecule. So they are kind of considered as junk DNA because they are not coding for anything specific, and that's making up 98.5% of our DNA. Is considered this junk DNA, which are introns. Despite its nickname, however, introns may function in gene splicing and therefore may have an important role in biology. So it's not that we don't use introns. It's not that I can't use it to create my DNA fingerprint or anything like that. It just means that like they're not coding for a specific molecule. So it is your code. It is specific to you, but it is not coding for a specific thing to be made. So they are junk DNA, but it's still useful in our science. So introns are still very useful to us in forensics. DNA identification. So most of the human genome is the same in all humans because we have all the same parts. We have the same working eyeballs, same working digestive system, the same working skin type cells, like all of those things we need. We need heart cells, we need blood cells, we all need all that same stuff. So a lot of our genome, a lot of our DNA strand is coding for the same things. But some variation exists, obviously. So most of the variation in DNA is found in the non-coding segment. So a lot of our differences are in that junk DNA, where it's not really coding for something because we kind of have to be coded very similarly. So not everything's like I have brown eyes, you might have blue, so that's gonna be a different code. But the fact that we both have eyes and eye cells, that's a lot of codes that have to be the same. So much of the non-coding DNA is the form of repeated base sequences, and some of these sequences can be repeated many times. So that's kind of what we end up looking at is in that junk DNA that isn't coding for a cell, but it still has a specific code like AATTGG, right? In that specific code, it might be repeated 5 times in your body, but 7 times in someone else's body. I did 10. So 5 and 10 times in someone else's body, and that's what is going to differentiate your DNA from theirs. Polymorphisms are the non-coded DNA segments that contain unique patterns of repeated base sequences that are unique to individuals. So our non-coded DNA, polymorphisms, that contain unique patterns of repeated base sequences. So it's like AATT, and it does it again, AATT. And does again, AATT. So that repeated sections of code. They don't code for a molecule, but they are specific in that you have that code in your body. For repeating DNA sequences, DNA fingerprinting isolates and analyzes the polymorphisms. Hard word to say. DNA fingerprints appear as a pattern of bands. Within the non-coding sections of DNA are short sequences of bases repeated multiple times. So the number of times that sequence repeats varies between people. So if we look at this example here, okay? So I have a code CTA, 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 CTA. It's repeating 5 times. But in the second man, they look for that same code and they find CTA being repeated, but it's repeated 6 times. And the third man, they still look for CTA. They're looking for that same code, CTA, 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 but this time it's repeated 7 times. So that is what different is. Like they both have the code, but it's repeated a different number of times. So example, if the repeated sequences CATACAGC, okay, so that's a long code, but if that's what we're looking for, we're going to look for that exact same thing to be repeated over and over down your DNA strand. One individual might have that same thing repeated 3 times, like this one, boop, 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 repeated 3 times, whereas another individual might have it repeated 7 times, making them have a different DNA sequence than you. The two types of repeating DNA sequences are variable numbers of tandem repeats, or VNTR, and that means they're 90 to 80 bases long. So when I go back to look at this, it's like this is 1, 2, 3, 4, 5, 6, 7, 8, 9. It's only 9 long, so that would actually be this one. That'd be VNTR. So the shortest it can be is 9 long, up to 80. That's a long code to look for. Short tandem repeats, though, so looking for a shorter one, is only can be 2 to 5. So the one that I previously showed you up here, the CTA, that would be an STR. I'd be looking for short tandem repeats of just repeating CTA, CTA, CTA. STR is much shorter and therefore is preferred for DNA fingerprinting. It's easier to do, faster to run. VNTR and STR data from DNA fingerprints can be analyzed for 2 main purposes. Tissue matching. Okay, so we're trying to compare the DNA evidence from the crime scene with DNA taken from the suspect. Kind of obvious. It's kind of our main goal. Obviously, in this class would be matching our tissue found from crime scene and our suspect or victim. So how it looks, 2 samples that have the same band pattern are the same person. Now they have to be exactly the same. So in the exact same spot, the exact same thickness, all the exact same band. So they can't have anything different at all. So this crime scene sample here, missing this, so it's not this person. Okay, matching, 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 matching. Okay, matching, 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 and it matches. Okay, so it matches suspect 2, doesn't match the other one. Now, again, does that mean that they did it? Did they commit the crime? Not necessarily. It means that they were at the crime. So it proves that they were there and they left their DNA there. But we have to, keep looking and digging to figure out if they did it. For this one, inheritance matching, so figuring out if they're their parent. So I mentioned this yesterday. Let's say you have a missing person. The only way to figure out who that person is would be to take our missing person's DNA, or if they don't have dental records. Dental records is faster. But if they don't have dental records and they're too decomposed to match to a picture, then we have to take their DNA. We compare it to missing persons that we think we know their parents. Okay, it's like this is a missing person that looks to be the same height, weight, sexual, sex of male or female. So we think this might be our missing person. So we go to that missing person's parents. We take their DNA. Okay, take their DNA, compare it to our missing person's DNA, see if they match. So comparing a family member's DNA for proof of familiar relationships could be for multiple reasons, but that would be one of them. So let's say this is the mother, the father's over here. So the mom matches, dad matches. So you're like, could be, sure. For this one, mom matching the dad, yes, could be. For this one, mom matching, this one, dad matching the dad, okay, could be. Matching the dad, matching the mom, could be. So basically, it's just all you're going to look in the child's row and you're going to see, do they have any band that doesn't match one of the parents? It has to match one of them. If it doesn't, then they're not their parent. So how it looks, each band in a child's DNA fingerprint must be present in at least one of the parents. 50-50 is usually about it. It's not always completely 50-50 in your quick DNA fingerprint. It should be about 50-50, and they shouldn't have any bands that don't match one of them. So quick practice. What can we tell about this family relationship? So here's our mom and dad, and they've color-coded it, so it's easy to see. But this one matches mom, this one matches dad. This one matches dad, this one matches mom. So that's their kid. This one, not matching, matching the mom. Not matching, matching the mom, matching the mom. So we're going to say, is the mom, dad's not the dad. This one, matching everything, that's their kid. This one isn't matching mom or dad. So neither of their kid. That have to be an adopted child. So neither mother or father is biological. This one, the mom's biological, but not the dad. And last slide, often the amount of evidence left at a crime scene is very small and therefore considered to be trace evidence. A problem with analyzing trace evidence is that many forensic tests will destroy the evidence sample, as is the case with DNA. So therefore, prior to doing the testing, sometimes we do PCR. I'm going to talk about that more later. So we're going to talk about the issue with trace evidence more tomorrow.